Video 3 for pre-location barn owls is about neuroplasticity and how you can use prism goggles to both reveal that this occurs and to study it. We're going to talk about how plasticity is age dependent and reversible in some specific cases, and then the mechanism for plasticity. So we're going to talk about long-term synaptic potentiation and then briefly discuss synaptic depression. So we've mentioned already why we study owls for understanding neuroplasticity. And this is a picture of a chick who is wearing prism goggles. And this, as turns out, has been a really powerful way to study how brains respond to mismatches between acoustic and visual space. And I'm going to start with a video that I'm not sure I'm going to be able to fast forward. So it's a few minutes, it's entertaining, and it's an example in humans.
This next guy is from Canada. He is Mike.
Okay, I have no idea what just happened there. Actually, I'm going to play it in PowerPoint so I can fast forward it real quick. Okay. Okay, so what he's basically going to do is he's going to ask a volunteer, I'm really glad I did this way because it's a long video, to wear prism goggles. And then he's going to do a short demonstration of learning, essentially. And this is in the case of kind of short-term learning. It's a little different than what we're going to talk about in owls, but it's still a good example.
And what we've got is well, we wouldn't call these lenses. What do we call these? And we've got right-angle prisms. Most people think of equilateral prisms, the one that make the nice colors. These don't make nice colors. You don't get the nice colors. What these are going to do is these are going to bend the pathway of light. We've got one side covered up completely. Otherwise, you get a little bit of internal reflection. So what this should do is move the pathway of light. So what I'm going to get you to do is put the glasses on. There's elastic there, but they're still kind of heavy, so you might have to hold them up with one hand. And I'm going to hold up my finger, and we're going to play the same game again. Touch my finger with your finger. Yeah. What you'll see is his eyes are not working properly now. We're going to keep playing this game, so I'm going to hold it out again, keep going. Hopefully, he will get better. And while he's doing that, I'm just going to keep doing that. I'm going to talk to them. You focus on.
So he did this well before I meet him.
The first experiment, so we're done with this. We're actually done with chickens. And someone made little chicken glasses and put them on, and go slowly, see if that helps. That's a little, keep going. And they put the food down for the chicken. If he does it twice, if not, don't give him false encouragement. See, I messed that time, so all people possible. These chickens, put down the food. Whoa. And of course they miss, like handlers. And just like handlers, they began to compensate. They learned to adapt. So what's happening just now is he's learned that instead of seeing my finger where it is, it looks like it's moved to the side, so he has to compensate. And that's what he's doing. And he's doing that very well. We're going to stop. Keep the glasses on. Don't do it. Now, what happens, of course, is now that he's adapting, he's learned to function with those glasses on. When he takes them off, he's still going to think he has them on. And he's going to continue to compensate even though he hasn't got them on. That's what should happen if things are normal. It doesn't always work that way, but we'll see. So we're going to do it, say, 3 more times with the glasses on. And I'll get you to take them off. And when you take them off, I want you to do it quickly, and wherever your hand ends up, we'll leave it there just so they can have Okay, so go slowly just now, so you're nice and adapted. There we go. One more time. Okay, glasses off. Quick. If you put this right side again, you can put it back down, go fast. He's missing on the opposite side. He still thinks he has them on. Now, he will stay like that for about 24 to 48 hours.
Okay, he actually figures it out quite quickly. So I'm going to go back into Keynote so I can draw when needed. Okay.
This next guy.
So there was an example of basically adaptation and learning. And so he was able to do that pretty quickly through a lot of conscious thought and effort. If you do this on a longer term, there are actually neural adjustments that take place and allow you to compensate without conscious thought and effort. And so we're going to return back to this example of when you plug the ear. So remember that when you plugged the ear and that the owl oriented the head to the wrong space in elevation and in azimuth, which we talked about in the last lecture. But what Eric Knudsen found is that if you leave the plug in the ear for a month, then the birds could actually then adjust, whereas adult owls could not. So only the young chicks could do it. The adult owls couldn't. And the ability to adapt required visual input. If you put goggles that were not, that you couldn't see through, then they couldn't ever learn the new location to look. So it required visual input. And so here's that experiment. So you have the first graph showing before the prisms, where the visual and the acoustic, the acoustic signal and where the bird turns its head to put its eyes are matched. But then you put on prism goggles that are 23 degrees shifted, and now all of a sudden the acoustic cues the response to the acoustic cues are correct, but the visual response to visual cues are now shifted by 23 degrees. So these are two different signals, essentially. You're either playing an acoustic pulse or providing a visual cue, and they're looking in different places is essentially what's happening with the head. By day 42, the orientation of the head is now adjusted so that the auditory, when a sound is played, they actually have shifted how they're looking, where they're putting their head, so that the visual cue maps on, the auditory cue maps onto the visual space. If you take the prisms off, then you can see that's flipped. So it's exactly what happened to that guy. You know, the person had their finger out and he was supposed to touch the finger. And with normal visual cues, he could do that very well. As soon as the visual cues were misaligned, he was touching, you know, he was missing it by several inches in the wrong direction. That's the amount of visual shift was evident by where he was trying to put his finger. The same thing you can do with auditory cues in owls. So when you play a sound, the owl looks to the sound. The prism goggles will cause the object to still be 23 degrees wrong compared to where the auditory stimulus told them to look. So they look at the sound, but the visual stimulus is actually 23 degrees over here. And so over time, if you play a sound still in that same space, they'll shift where they orient their head so that they're looking now at the visual cue as well. So that's what that experiment did. So visual and acoustic place information merge in the optic tectum, which I mentioned before. Here's that drawing on the right, where you have the two pathways for interaural level differences and interaural timing differences that merge in the inferior colliculus. That's this structure here. There's the inferior colliculus. The ICX, one particular part of the inferior colliculus, is where you get really strong place, elevation, and azimuth responsivity. And then the ICX, the ICC in general, are sending projections to the optic tectum. And this is where there's an overlap between the acoustic map created by the ICX and a visual map that's created in visual processing centers. and they're overlaid so that they match normally anyway. And so what you have on the left there is an example of when it's normal. The auditory and the visual acoustic space align. And so if you have a flash of light or a pulse of sound coming from that particular area in three-dimensional space, then the head will always orient to the same place. If you put on the prisms, then now the auditory cue from the same exact spot will cause us, you'll have a sound. But when they look at that place, the visual system has been shifted by the prism, so it's not matching anymore. After 8 weeks, then you get a realignment, and what's happened is that the acoustic map has shifted to match the new visual map because of the prism. Okay, so the acoustic map somehow shifts. So if we look at this, just in another example here, this little drawing I drew, you start on the left, and if you have a sound directly in front of the bird, then you have a 0 interoral timing difference. And you have no prism shift, and so you have alignment between where the sound originates and the visual cues that are at that place. So let's say that's a bell. So you see a bell. When you put on prism goggles, now you have an unnatural situation, right? So if you put a sound directly in front of the bird, it still has a 0 interoral timing difference, but when it looks there, the bell's not there. The bell is now shifted 23 degrees in this example to the left of the bird. And so when it looks at the bell and the sound, there's no visual feedback that says, yes, that was a correct head movement. Instead, the visual feedback is shifted a little bit. And so what's going to happen over time is the bird's going to use that visual feedback to say that was wrong. And what the head really needs to do is when it hears a sound, it needs to shift where it looks in order to map it on to the new prism shifted visual space. And so basically now, in order to look at the object that was previously straight in front of the bird, so it's still straight in front of the bird, if you imagine the body below it, the body's position hasn't changed. But what it needs to do now, in order to see the bell in its visual space, is it needs to look 23 degrees to the right, and that will cause it to map on to the correct space. So what it needs is for to have that head movement assigned to a 40 microsecond interoral timing difference instead of 0. And then it will cause realignment with the visual field. So here's what happens in the brain to neurons before, during, and after the prism shift. So this is an optic tectum neuron. So this is where the visual and the acoustic maps are overlaid. And what you see is that you have a normal interoral timing difference, and this optic tectum neuron responds really well to it. The learned ITD, I'm going to just point here with so here's the normal ITD in the optic tectum. That's before you put any prisms on. And there's low responsivity to what will become the learned interoral timing difference once the prism goggles are on. But right now, that hasn't happened, of course. It's just this normal interoral timing difference. You put on the prism goggles, and after a certain number of weeks, you start to see that the response to a 40 microsecond delay, okay, this one over here, this 40 microsecond interoral timing difference is starting to show some responsiveness. So that same neuron that used to not respond to that timing difference is now showing a much stronger response. And then interestingly, after a full six to 8 weeks, you see that the responsiveness to the original correct interoral timing difference has now been inhibited. We're going to see why. So the interoral timing difference maps, when they did this experiment, interestingly remained the same in the inferior colliculus, but it shifted by the time the central body of the inferior colliculus, it shifted by the time it got to the ICX, the external part of the inferior colliculus. and then onwards to the optic tectum. And so the proposal then was that there must be, so on the left, that's the normal. Okay, so you have a place specificity in developing in the ICC that corresponds to a particular interoral timing differences. So these are all basically the delay lines are built in along here. Okay, so you've got the right ear coming in, the left ear coming in. So that's how you're getting these interoral timing differences. And then those are projecting to the ICX, where you're getting this auditory space map. And then onto the optic tectum, where you're getting a mapping onto the visual field. And so here you have 0 microseconds is equivalent to 0 degrees in visual field azimuth. So a sound and a sight directly in front of you excite the exact same place in the optic tectum. And here, a 50 microsecond interoral timing difference corresponds to a 20-degree visual field shift. So, and those are going to map on perfectly. So a bell that's 20 degrees shifted provides a 50 microsecond ITD and a 20-degree shifted or 20-degree visual field. When you put on the prism goggles, it changes the relationship between the visual azimuth and the auditory or the acoustic azimuth. And so what you see are these red lines here in the right-hand drawing that are shifting the relationship. This is the plasticity. Okay, you've got the old ones, the gray ones, that are underlying there. I'm going to draw them in white here. No, not white. It's not going to be a good color. How about yellow? So here's the old one. The new one has formed synapses and shifted where it synapses from the ICC to the ICX. And that's going to allow that acoustic field to shift relative to the visual field and now cause a 50 microsecond. So it used to be 0 microseconds correlated with 0 degrees shift. And now what we're seeing is that a 50 microsecond correlates with a 20-degree prism shift. Okay, it turns out that juveniles can do this and adults cannot. But one thing that's really interesting is that if you take juveniles and you put prisms on them, they will adjust. And so the left-hand panel A is the shift in the interoral timing difference relative to normal. So it required a shift of 30 microseconds in order to map back onto the prism shifted visual field in this bird. And there's 3 different individuals there that are showed. So they all shifted their ITD in order to match this new reality that they had in their visual field. If you take those prism goggles off, it shifts back to normal. So they again map a 0 ITD with a 0 visual field. What's cool is that even if you leave them normal for a year, if you put prisms back on them as adults, they can shift back to the prism adjusted values. Those are the red lines in B. So they're able, after about 20 to 60 days, to shift back to essentially where they were as juveniles when they were wearing prisms. But if you take adults that had never experienced that as a juvenile, they couldn't do it. And so essentially, they had to have had the experience as a juvenile to form those connections, those other weird, different prism-adjusted synaptic connections in order to have the ability to do this as an adult. So in other words, what we think is that if birds experience this as juveniles, then as adults, those connections are still there. So they're not lost. They're there, but they're not active if the bird's not wearing prism goggles. They can become activated if the bird is wearing prism goggles, but only if they were established during development. So what that tells us is that basically There's a critical period during development when this is possible. Okay, when this brain region, the ICC to the ICX, allows for some synaptic plasticity and allows for other connections to be strengthened or not. I wish I had left a blank slide here because, so I don't know if you've heard of synaptic pruning before, but when you're young, I'm just going to draw over the top of this. We'll draw an ICC and an ICX here. When you're young, there are connections. So let's say we're going to do connections from like one neuron in the ICC to the ICX. There might be, its axon might spread out to that many places on the ICX. And through experience, those connections get what's called pruned back. In other words, the synapse first becomes silent, so there's essentially no communication between this neuron here and, we'll do numbers. So let's say it makes 6 connections along there. And it learns through experience that the best match is with 4. Then eventually the synapses, these axons that connect to 1, 2, 3, 5, and 6, will die over time, but only through inactivity. So if they're not used, they will eventually, as an adult, not exist anymore. A will only connect to 4. There will no longer be, even though these neurons are still there, the connection now is only from place A to place 4. But if the bird is exposed to prism goggles when they're young, and let's say that that prism goggle correlated to here instead of 4, then when they're older, that connection, because it was utilized as a juvenile, will be maintained. And so it may not be active, and it may be actively inhibited in a non-prism wearing situation. But essentially, because they experienced it as a juvenile, there's now the capacity to do it again as an adult, because that pathway was potentiated as a juvenile, and therefore it was never pruned away. So I use this word potentiation, and that refers to this concept of long-term potentiation, which is the persistent strengthening of synapses based on recent activity. In other words, lots of strong stimulation increases the sensitivity of the synapse to excitatory neurotransmitter. And it's implicated in forming memories, it's implicated in learning, and it's implicated in plasticity. And so this involves 2 different glutamate receptors. So glutamate is our main excitatory neurotransmitter in sensory systems. And there's 2 different kinds. So one is called AMPA. So the AMPA receptor is ionotropic. It's a ligand-gated sodium channel. So when glutamate is present, it binds to AMPA, and AMPA opens and allows sodium to enter the cell and depolarize it. NMDA is also a ligand-gated channel. It also binds glutamate, but it allows calcium to enter the cell. So that's one difference. It also has a second gate, essentially. It has a magnesium plug, and it requires depolarization of the membrane to open that gate, it's effectively a voltage gate, and allow for calcium to flow. And so in the first step, you get repeated excitation of AMPA. If you get repeated excitation of AMPA, then you get a large enough membrane potential from the sodium flow in order to open the NMDA channel. And then calcium influx comes in, and that starts a second messenger cascade that effectively strengthens the synapse. And it does that by putting more AMPA into the membrane, so that in future events, if you have more of the ligand-gated sodium channel, then when a presynaptic cell releases glutamate, it's going to respond even more strongly because there's more receptors to open up and allow sodium flow. We're going to draw it. Okay, so we'll draw a presynaptic neuron that has an action potential coming down, and it's got its vesicles of glutamate. And I'm going to draw a bigger postsynaptic one so that we can draw. And it's got AMPA receptors and NMDA receptors. And so when this presynaptic cell fires an action potential, you get glutamate release. And glutamate is going to bind to AMPA, and it's going to bind to NMDA. Glutamate binding is enough for AMPA to open. And sodium is going to flow into the cell. Now what happens next is dependent on how much glutamate is present, how much AMPA is in the membrane. But basically, if you have only a few AMPA receptors, then maybe there's not enough sodium that enters. I'm going to draw something in the. Okay, so if you get a little bit of sodium entry, then that might not be enough of a depolarization event to kick out that magnesium plug I just drew in the NMDA receptor. The NMDA receptor I mentioned has both a ligand gate, that's the requires binding of glutamate, which it has right now, but it also is essentially voltage gated, and it's voltage gated because this big magnesium ion sits inside the channel. And the cell, which is normally negatively charged, right, at rest, the VM is negative, and so it attracts that magnesium plug, and the magnesium plug doesn't get ejected. It's on a little tether here. If you have, so we'll just say this. This is sub-threshold. So in that scenario, you didn't get an action potential or a NMDA activation. But let's say there's twice as much AMPA. And therefore, when there's a glutamate event from the presynaptic cell, you get twice as much sodium flow. So here you got a little bit of sodium, here you get a lot of sodium, and the VM reaches threshold. That may be enough so all this positive charge coming in around the NMDA receptor might be enough to kick open this magnesium plug. In other words, the positive voltage building up on the inside of the membrane expelled the positive magnesium ion and kicked it out of the channel. So now what happens is that NMDA is also a cation channel, but it's specific to calcium. So calcium floods the cell. And calcium essentially binds to calmodulin. We'll just gonna give the abbreviation there, calmodulin, and that's going to trigger a bunch of cellular responses that eventually leads to gene transcription. And that gene transcription is going to cause the insertion of more AMPA. So basically, by heavily exciting a synapse, the synapse, if it's equipped with an NMDA receptor, will cause a calcium influx that will eventually cause even more insertion of AMPA receptors, and so you get a strengthening of the synapse. More AMPA equals what's called a stronger synapse. Because now it takes less glutamate to reach threshold. So hopefully that makes sense to you guys. That's called long-term potentiation. In owls, if you give ketamine, which is an inhibitor of that NMDA channel, in other words, it prevents it from functioning normally, it prevents it from opening, then you also inhibit the ability to learn a new response. So if you look at activity in the OCX, you have the normal responses here, and then learned responses here when there's no ketamine present. So that's the control. And then when you have ketamine present, the normal responses drop a little bit too, because ketamine is an anesthetic, and so it's inhibiting activity, generally speaking, but it inhibits much more the learned responses. So it really prevented learning. And it's thought that because ketamine has a strong impact on NMDA, that this is evidence that NMDA channels are involved. And in fact, research since this paper has supported that idea, although it's incredibly complicated. In reading some of the newer stuff, it basically goes from this to I'll just say extreme complexity in terms of what neurons are responding in what way to all the different kinds of inhibitory signals, excitatory signals. It's really complicated, but it's also very cool that people are starting to get into the nitty-gritty of how all this happens. Right now, we're going to focus on the broad concepts. Okay, so what about inhibiting connections? We know that in these complex center surround systems, for example, you have inhibition. We know that in learning, so we had those, the connections. Let's see, how far back do we have to go for that? Here, so we have the red connections that are the prism shifted, but then there's still also the gray connections that exist that were the original connections between the ICC and the ICX. And so we saw that you had to both excite the new connection at the same time that you inhibit the old one. And so essentially that's the opposite of what we just saw, where a lack of stimulation, so the longer a synapse sits silently or with very little activity, the less AMPA the cell is going to put into the membrane. And so over time, you go from having kind of a normal synapse that could respond to glutamate to having a either depotentiated or a depressed or a silent synapse. And those terms are basically, so you have your synapse and you start out with maybe lots of AMPA. If you've had recent long-term potentiation, and then you'll get depotentiation, which is here, back to kind of a baseline state. I'm going to switch to their drawings. I'm realizing I'm drawing the same thing I put in there. So you have, say, a high density of AMPA. You get depotentiation, where basically the cell just stops putting as many AMPA. And as the AMPA get recycled through kind of normal cellular processes and renewal of the membrane and whatnot, you get a reduction in how many AMPA there are if it's not an active synapse. Activity at the synapse would potentiate it and maintain the number of AMPA. But so that's going to drop. It goes to what's called a depressed. So this is IT, sorry, LT, I'm used to IT. LTD Long-term depression happens when there's actually removal even further and you go kind of below a baseline state of AMPA receptors. And a silent synapse, this is what would happen right before a synapse might be pruned. Especially during that early development, if it wasn't being used at all, because that's one way for a synapse, a neuron to know, okay, I branched out and made 5 connections in these different regions in the ICX. The only one that's being used is synapse C. And so over time, all the other synapses are going to be eventually retracted. The axons are going to retract, and those synapses won't even exist unless they get used somehow. Okay, so when a synapse is depressed, it makes it more difficult to a cell to reach threshold, even when there is glutamate present. And so this is one way to enhance or inhibit connections between cells. Okay, so I'm going to end. I tried to slide there. End on these artistic versions of long-term potentiation. I really like science art and the art of just, communicating science. And so I found these when I looked up long-term potentiation and art. So I'll leave those for you guys to explore. And I'll talk to you guys on Tuesday.